Swayam Prabha. Digital India. Educated India.
Okay, so now I told you all about how to see inside the cell, right? So now all those pictures that you see when it comes like, viruses and all that is by electron microscopy. You do 3D reconstruction, you do analysis of the interactions. Everything that you do generally will be by electron microscope because you want detailed information, right? So there are multiple components that you can see with this. You can see membranes, you can see organelles, you can see ribosomes, these are machines which produce proteins. You can see something called as filaments. So our body has a skeleton, right? Like we all have bone skeleton, which keeps us in shape and which gives us a form. If I want to move, I can move my body because I have a rigid cytoskeleton. I have a rigid skeleton, sorry. So what about cells? Do they have a skeleton like that? Or are they like, they are squishy? So how do they move then? That is inside the cell. For movement, there is motor protein. But if the cell itself wants to move, how does it move? Yeah, there are different ways for how it can move. So cells can move themselves also, sorry. No, there are cilia and flagella which is used for propelling it, but the cell itself can organize like amoeboid movement and all and move further, right? So in a cell, there are different aspects that are important for the size, the shape, movement, right? So when you look at a cell, this is how you'll see generally, like different kinds of cells will have a filamentous like backbone. Like there is something which is present in the whole cell. There is like a network that is present which holds the cell, which helps it move. As I said, there is transport within the cell. All that happens based on this, okay? They will also move. So if you, what you said, other than cilia flagella also, you can have movement of cells. So they will put one part of them outside and they'll move because they want to move inside your body here and there. So if you put them on a slide, they will move around here and there. So you will see that kind of movement. Here you can't. So all of this is brought about by the cytoskeleton. So this is in some fibroblast cells and this is within a neuronal cell. So within the cell also, there are small things which will move. So the cell moves, things inside the cell moves, and it is all maintained. Okay? All of this is done by something called as the cytoskeleton. As the name suggests, it is cytoskeleton. So skeleton of the cell. Okay? So it is important for size, shape, structural integrity, cell division that you see. All of that is brought about by the cytoskeleton. Now generally, the cytoskeleton has three different kinds of filaments. Okay? You have actin, which is also called the microfilament, and that is like a thinner filament. Now we are talking about nanometers, okay? This is all within the cell. So we are talking in the range of nanometers. So you can call actin filament as a thin filament here in terms of the other filaments. And then you have the microtubule, which is 25 nanometers thick. This is like a tube. Okay, I'll talk a little bit more about each of them also. And then we have intermediate filament, which is between them. So all these three are involved in the cell for different purposes. You want a thin filament for more flexibility when you want it to organize faster. You want a thick filament when you have to do a lot of force work. So if you know in cell division there is spindle formed, right, for dividing the chromosomes. That is formed of microtubules because these are very big DNA molecules. You want to divide them, right? So that you will need a very strong polymer. So that is done by microtubules. Then intermediate filaments have a different properties, like your hair, keratin, that is an intermediate filament. That's a polymer. It is less dynamic as compared to these two. So you have different properties for that, more structural properties and all. But all of them together make the cytoskeleton, okay, along with other proteins which also bind to this. So I'll very briefly tell you some of the properties of the cytoskeleton, yeah. So one is they are dynamic polymers. So for example, actin, it is made up of, so these are not, so when they are produced, they are not produced as a filament. You have one unit which is called the globular actin or G-actin, and then there is polymerization when needed. So now when the cell wants to move, it needs to make all these filaments which will make the cell move. It will provide the skeleton, right? That's when it will synthesize all these filaments. Otherwise, inside the cell, it is present as G-actin. We call that globular actin. So you have a whole curve, how polymerization of this filament happens. And then even in terms of microtubules, you will have similar. So you have tubulin, which is formed. So you have two of them. In actin, you have G-actin or globular actin. So this is the monomer. And then it forms a polymer which is called filamentous actin. So this is the polymer. And then in microtubules, which is the other dynamic cytoskeleton, you have tubulin, which is the monomer, and then it forms a polymer, which is the microtubule. Okay? So you have basic building blocks which will form them. Usually this will be mediated by some energy process. So here you have ATP hydrolysis, and here you have GTP hydrolysis. So it's very nicely built in the system. You have two different molecules that will use for two different filaments. So when you want to do cell division, GTP will make microtubules, it will help in cell division. When you want the cell to move, it will make actin, actin will help in movement. So it's very nicely compartmentalized and organized. Oh, sorry. Okay. So this is adenosine triphosphate. GTP is guanosine triphosphate. So it's a different, there's a small change in the ring that you see, and it's a different one. So it also produces energy when there is hydrolysis of the bond. But it is, I think you all may not be familiar because it's not used so often in other processes like. Yeah, so you will need it. So ATP is the more common energy currency of the cell, but GTP is also used, and that is used in tubulin to microtubule. So it's nicely compartmentalized in the cell. So when you read about this, you can also think about how they are all sitting inside in the cell, right? Like how do they work together as well as separately. You don't want actin and microtubule just to be produced inside the cell. They are produced when they are needed and when they are formed, right? So that way it is controlled. So polymerization will happen like this, like, you have addition of units, and then in case of microtubules, they sometimes can destabilize and fall apart. Actin doesn't fall apart so easily, and intermediate filaments are a little bit more stable. So their properties are also different. So how you want them to behave in the cell, it all depends on what is being added. Like, so yesterday you would have done some microscopy, I think, fluorescence microscopy, right? So you can use fluorescence microscopy to monitor this kind of polymerization dynamics. So I am showing it outside a cell, but you can also see it within a cell, like you can see some images. So here what I'm showing is I have one microtubule which is labeled in one fluorophore, which is a small set of microtubule I can keep, okay? And then I add tubulin of another color labeled. So I've added a dye to tubulin, okay? And then you can monitor how it is growing, how it is shrinking. So you can measure the rates of polymerization, depolymerization, and you can do this under different conditions and all. If you want to study, for example, migration, is there an increase in the polymerization rates of microtubules? Is there a change that happens when you're doing some other process? You can do that measurement by doing this kind of microscopy experiment. You can do that within the cell as well. Yeah. So they are very dynamic polymers which you can see or measure the rates of their different properties. Another thing that is important is you might notice that they are organized in a particular direction. So these are polar polymers, which means they have one growing end and the other which is either slow growing or non-growing. But there is a kind of directionality. It's not the same in both sides. Like not exactly this chalk, but if imagine it was a cylinder, whether I look at it like this or look at it like this is the same, right? So these are called as, they don't have directionality, the polymer. Whereas if I'm looking at this bottle, so whether I'm looking at it like this or looking at it like this, it's different, right? The polymer or the cylinder is different. So that means that it's a directional polymer. So this will be important. I'll tell you why it is important. The direction of the filament is also important because that will give the force. So where you want it to grow, it will grow in that direction. So you want the cell to move in this direction. There will be addition of polymer in that direction. You can't add it like this. It will provide a reverse force then. Photo, yeah, so it will be in one direction. So there will be always a directional movement. In this case, it is a simpler process.
Can we compare it to the DNA replication? Everything comes in the nucleotides that are added to the 5' end.
Yes, so that will also be. So it will be only in one direction. So all this is important in cellular processes, yes.
What causes directionality in the cell?
Yeah, so that is because the building block is not symmetric. See, if I had a building block like a circle, I added it here or I added it here, it wouldn't matter. But this building block is like this. So now if I add it here or add it here, there is a difference, right? How it looks. This side is flat, this side is pointed. So the building block is like that the way it organizes, one direction looks different from the other direction. So that's why there is directionality in this point. And because of that, I'll tell you the proteins, that motor proteins also change their direction. Yeah. So the other, yeah. That's how they are built. So that is a way. But you can think of it. So it's not a why in the sense we don't know the reason why it would be. But if you think of it, this is the design of the cell. And it will help you in, like I said, giving a directionality. Like otherwise, if you had just tubes inside the cell, how will you make them grow in one direction only? Like imagine you have a cell, right? You have a cell, this is the nucleus. I want it to move here. You want something to give a force. There is a cytoskeleton here. You want it to give a force in that direction. How will you do it then? If you just had a tube, it will grow in both directions, right? Because if it was just a circle, both sides will have the same propensity to add another monomer. But because it is directional now, I can orient my molecule and then I start polymerizing there. So then This is a guess. I don't know. This is maybe evolutionary. It was designed like this to give this kind of a purpose, right?
Can you also orient this cylinder to like grow like this, like on
So,
an IRL that?
yes. So I will just show you. So generally, how it is regulated is by interactions between this. So when I'm adding this. Okay, so when I'm adding, okay, let me label. So when I'm adding A, B, and C. So when I'm adding C to B, this interaction is more favorable than when I'm adding A to B. That's how we make it directional. So it's going in one direction. And this is a better interface. So if you are adding on the side, that interaction, if it is favorable, you can make that. But I'll show you in microtubules, you have that also. But I'll just show you that. But generally, this is the most favorable interaction. So you proceed in that way, like up. So as you said, like within the cell, you have different kinds of arrangement with this filament. You just don't have single filament. So for example, in actin itself can form like one polymer, or you can have branches also. There is a way to form these branches. There are other components which help in this. So there are other proteins which will help it in branching. Also, like I've shown here, with different other proteins, you can make different kinds of networks. You can make parallel network, you can make crisscross. And what happens is the properties of the filaments change because of that. So when I have a bundle, you can imagine, you would have all read that story, like 10 bundle is stronger than one bundle. So if you want to make like a strong structure, you will bring lot of these actin filaments together. It will make a strong structure. If you want a jelly-like surface, you want like protein to move around, you make it like this, like loose crisscross. In the end, I'll show you a video where all these different things come together. So there will be like crisscross. So depending on how you want your skeleton to be, you can change. So you give them one component and then change all the other organization. So like that you can do for microtubule also. So with only this one building block of tubulin, what you said, you can build it on the side. So in cilia and flagella, so apart from spindle microtubules, in cilia and flagella, have you all heard of the 9+2 arrangement? Yeah, all that if you have a cross section, you have 9+2, no? So each of that 9+2, so you have cross section of a cilia, no? So each you will have 9 like this. So each of this, we call it a doublet. This is actually 2 microtubules. So one microtubule and another microtubule, half microtubule fused, that forms a doublet microtubule. So that cross section wise, this is how it will look like. There are a lot of other proteins, but you have like these different kinds of structures which you can form with microtubules as well. Yes. Yes. So in cilia, do you know cilia and flagella? Like you have these small appendages on a cell which help movement of the particles on the surface, the fluid on the surface. Now when you look at the cross section of a cilia, it will have this kind of arrangement. I'm not drawing to scale. How many did I draw? Correct, right? Okay. So if you have cross section of a cilia, this is the membrane, and here you will have this kind of a framework. So actually I've done some work on cilia. So cilia it's a very, very interesting structure. So it moves a lot, like you know that movement it has, no? And it has to be very stable when it does this. It should not break, you know. These are all just proteins, like I'm just putting like a chain. Sorry? So cilia, right, so cilia is smaller. So you have to realize that flagella also is different in bacteria and in eukaryotes. So we also have cilia and flagella, which has this kind of an organization. So if you have a cross section, so as you said, cilia are smaller in number, they are more numerous, whereas flagella you'll have one. So like parasites and all, if you know trypanosoma and all, they have a flagella that will be built like this. But bacteria have another flagella that is built of flagellin protein. That's another protein. That doesn't have a 9+2 organization. That is a motor. That is what you need for moving here and there. So the movement of bacteria is different. It does this movement like this and goes behind and all that. Flagella in trypanosoma is different. So they have a waveform flagella. I don't have videos now, but their movement is different as compared to bacterial flagella movement. So they are all, so when you think of it, they are all built differently for different function. What their function is, for that they will be built accordingly. So this 9+2, if you imagine, so this is a cross section, so it will be like this, like there's a cilia like this. So this whole is a tube, like here it's a tube. I think later on maybe I can show, on my laptop I have some figures where I can show you. So each of this is a microtubule. This is one microtubule and this is half microtubule, which is added. So it gives a lot of structural stability. You can imagine like 9 of them together. So when the cilia and flagella is doing its job, it doesn't break or it doesn't like, collapse or it doesn't do its job properly. If there are any mutations in any of this, then you'll see diseases because the cilia cannot do its job properly. So that is one way of this. So this is the 9+2 arrangement that we were talking about. Like it is made up of doublet microtubules.
If you're counting each of those pairs as one,
Oh, no. So this is not a doublet. The center one is not a doublet. They are two different, they are called as a central pair. So this is like C1 and C2. They are single microtubules. So they have a different organization. Oh, I don't have a cilia here. So they have a completely different organization. So they are linked like this, like this, like this. They have proteins which link it. That's why we call it 9+2. Yeah, so that's why. So inside the cell, using a combination of both the filaments and the other proteins, you can make a large number of structures. So all kinds of structures that you see inside the cell will be a combination of different of these proteins. Okay. So, yeah.
Does this tubular protein structure also occur in a centriole?
Yes. So centriole is the third one. So this, okay. So you have, this is all what I work on. So I'm very interested in understanding all these. Yeah. Huh? Okay, so centriole, it's okay. So centriole is, so inside a cell, when you want to divide the cell, you have these two spots which come up on the end. Okay, so that is centriole. But what is interesting is the microtubules grow from the centriole. So that is very interesting. So what happens is you have one microtubule, like this is one microtubule, right? So 25 nanometer. That is, I'm just showing roughly. So you will have a doublet, we call this a doublet. This is what you see in cilia and flagella. In centriole, you have a third one. So you have, it's called a triplet. And it's very interesting actually, this 3 becomes 2, becomes 1 when it is growing, for example. So the base of the doublet, I think, I don't know, if you are not understanding this, let me know. So if the base of the doublet is also a basal body, which is like a centriole. So it goes from 3 to 2 inside every cilia and flagella. No, actually, it is a gradual transition. So inside the cell, there are, so these two continue, this one stops. These two continue, this one stops. Yeah, so we call it a transition zone inside the cilia and flagella. But it's quite interesting how it is synthesized. And inside a cell, you will have very short, like this centriole, and from the centriole, you have centriole and then you have pericentriolar matrix, and from that there is microtubule which is produced. I can show you all some videos and all of how it grows. There are these units from which the microtubule grows, and that will give you the spindle fiber for separation. But the basic idea is large number of structures with just this building block. So there are only two building blocks. There is globular actin and tubulin. From that, using a different combination of proteins and all, you can make like all kinds of structures. Yeah. So there are many proteins also which associate with them, which are stably present, which bind on these. So some of these proteins are static proteins that they bind and make different structures. As you said, there are some other proteins which are called motor proteins. So they actually literally walk on that. I'll show you all some animations where I'll show you some real experiments where you can see them walking on the microtubular actin. So with actin, you have myosin, which is a motor protein. You will all know the sarcomere. Are you familiar with the muscle, right? So you have contraction of that, right? Yes, so actin and myosin forms the filament disc. I mean, there are a lot of videos online, but so I didn't put them. So you have rows of actin, and in the middle you have myosin. So these myosin will pull the actin like this. So that's what causes that contraction. So you have actin filament like this, and then you have myosin head. And then you have actin, and then you have myosin. So these engage with the actin. So this is myosin, and this is actin. So these engage and pull the filament, and that's how you see the movement. So in actin, that's one of the major motor. In microtubules, you have two motors. We have kinesin and we have dynein. And I told you all that these are all polar polymers, right? Which means they have a directionality. So this is where it is important, and it's very interesting. Kinesin walks towards the positive direction, and dynein walks in the minus direction. So these, yes. So every polymer has we call it a plus and minus end depending on which end is more active in polymerization. So we call plus end is where more polymerization happens, and minus end is where less polymerization happens. So the kinesin moves in this direction, and the dynein moves in this direction, always. It literally walks. I will show you in the end a video of how it walks, but yeah. Yeah. So it's all about protein-protein interaction. So the way kinesin is built, it interacts in this direction, and it can move only in that direction. So how do I tell you? So if I'm thinking what is the best way of telling. If I want to go in this direction and I have put my foot here, the way kinesin is, the way the polymer is, it cannot go back. It has to go front only. The way it is designed. It is, so the way it actually looks like this. Yeah, it can only move in the forward direction. It sits like this. It cannot sit like this. It cannot interact like that with the microtubule. So it sits like this. And after that, it can only move like this. I'll show you how it walks also. It walks like this one. It literally walks. There is ATP hydrolysis. It will hold a cargo or some protein on its head and it will carry it. Dynein sits like this. Dynein cannot sit like this.
Why?
The interaction doesn't allow it. So,
So is there like a function of some sort now or an attraction that causes it to sit like that?
so we call it like a footprint. Maybe how I'll explain it is, so if microtubule surface is like this, okay, and like this. So, okay, let's make it directional. I'll just make it a little directional, just like to make it directional. So if I have a molecule like this, so kinesin is like this, okay, so it can only sit here. It cannot sit here because it doesn't fit. So then it will go in this direction because after that it can only move forward. It cannot move backward. Now I have dynein molecule which is like this. So dynein will only sit here. Dynein cannot sit here. And it is oriented this way. So then it will move in this direction. So the surface of the microtubule is like that. So we have structures of kinesin and dynein and all also. So you can see how the movement happens. There is lodging and then movement in the front direction. But it will always happen in that direction. People have done a lot of studies where if you put both kinesin and dynein and you give it a molecule, what happens? Like will it tear it apart or will it go in which direction? They're very nice experiments. So the answer is that dynein is a stronger motor, like in terms of its interactions and the amount of energy. It looks also very big. It has a lot of adapters and all that. So if I put one kinesin and one dynein, dynein will take it. Dynein will pull kinesin and take it with it. So it's very cool actually. So you have a microtubule. I put dynein. So this is dynein. I put some attachment on the top, and I put kinesin. This is kinesin. It will actually pull off the kinesin and take it in that direction. But what I can do is I can add more kinesins. If I add more kinesins, up to, there's a ratio depending on which kinesin you're working with, up to 5 or 10, you can make it pull this side. Then it will pull the dynein. Yeah, it depends on which kinesin. So there are many kinesins, they all have different properties as well. So depending on which kinesin you're doing your experiment with. So you can imagine in the cell how this is working. Like if you want to move it in one direction, which motor will come, other motor has to be inactivated. You ask it to move with this many things. So all that combination happens inside the cell. But that is how, if you imagine, transport happens within the cell. If you want like directed transport, I know, so my road, I know I'm going from Chennai to Bangalore, not Bangalore to Chennai. And I know I'm taking a truck or a car or a scooter, what you want. You take that accordingly and go. So that is how transport happens. Yeah. So this is one animation. Oh, it's not very bright. So these are, the red is the microtubule, and the dots are actually the kinesin motors. I don't know if it's, yeah. So you can do such experiments where you can see the motor moving on the microtubule with time. So you can see how the interactions happen. Okay, so I think I'll just end with this video where it actually shows the different filaments. I think it's working in a cut. Yeah. So these are like so inside a cell, you can imagine this is how it happens. So there are all actin filaments. And what you saw earlier was organized arrangement. Here it is slightly less dense arrangement because you want it that way. And so actin, they'll all come together, they'll polymerize. All this information we have gotten from mixture of microscopy and electron microscopy. So all these filaments will come together and they'll form inside the cell. This is a severing protein. if you want to chop this, suddenly you want to destroy all of them, you can chop it off like that. Like you can cut the filament inside the cell.
How does it break down? Severe protein.
So it will bind and kink the filament. It literally bends it.
Okay.
So then the interaction breaks. So this is a microtubule, and it will polymerize, depolymerize within the cell depending on what all proteins are there to stabilize it. And then that's a kinesin. So it literally walks like this. That's how it is walking. It will have a vesicle, or it will be, we call it cargo. It can be attached to an organelle. So it will walk on the microtubule it takes these steps. It takes 4 nanometer, 8 nanometer steps. There have been many experiments to validate this, how it walks on, takes one step and goes. Sorry? Yeah, they're pretty fast. Yeah, like microns, nanometers per second, microns per minute, around that range. So, yeah. So that's the centrosome, and from that you will have all the microtubules coming out. So yeah, I think, so that's about it. That's the general idea I wanted to give. And my work, what I have done in the lab, like this is one of the experiments. So just wanted to tell you all how I brought all of this together. So I wanted to look at microtubules or cytoskeleton inside the plasmodium, because plasmodium, I told you, it goes through different stages, right? And the shape and all looks very different. So when it is organized like this, it rebuilds its cytoskeleton at every stage. So if we are able to understand how it does that, it can be used to target drugs, it can be used to kill the parasite and stuff like that. So we wanted to understand that. So my colleague actually, the videos that I showed you of the parasite, she did that work to, she grew the parasite on that grid that I showed you all, and then you can image it and see how it looks like inside. So we wanted to get more information. So what I did was we got this plasmodium also has tubulin. So we purified the tubulin from the parasite. There's a whole procedure for that. And then you can, what I showed you is on the slide, it will start polymerizing. So in a microscope, you can see polymerization of these filaments. So you can add GTP and then you can see, so it has started forming polymers. So I purified tubulin from the plasmodium parasite, and you can see now it has started forming these filaments. And now this I can use as my sample for electron microscopy. So this I can put on my grid and then I can image it. So I did that. So this is how it looks like. Like this is an actual micrograph of microtubule from Plasmodium falciparum, which causes malaria. And then I can do the 3D reconstruction, which I showed you all earlier. You can do a 3D volume. So that's how it looks like. So this is, after I do a 3D reconstruction, this is a microtubule of Plasmodium falciparum. So you can get a lot of information from this. Like each of this is one tubulin, and we call this like a protofilament. So I then did analysis of how this interaction happens, what is different in us and the Plasmodium, if we can design some drugs that will inhibit this, that kind of experiments and all you can do. So I think that's all I have. That's basically what my work is and what we use for looking inside a cell, looking at the cytoskeleton. If you have any questions, feel free to ask me. I think our email contacts might also be there. So if you want anything, you can always write to me. So that's about it.
Swayam Prabha, Digital India, Educated India.